What if aging is not just a slow decline, but a process tightly governed by how our cells use energy, sense nutrients, and regulate inflammation? What if molecules like NAD+, even ketone bodies shaped by diet, metabolism, are quietly influencing how our genes are expressed and how we age? Today's guest has spent decades uncovering how metabolism, epigenetics, and the immune system converge to shape human aging and what that means for extending health span. Welcome back to Decoding Longevity. I'm your host, Dr. Raghav Sehgal, joined with my co-host, Dr. Maxwell Jackas, and today we're joined with Dr. Eric Verdin.
Hi, everyone. In our last episode, we have explored longevity policy and how to bring aging science into the public action. Today, we're going much deeper into the molecular biology of aging itself, from how cells sense nutrients, regulate energy, to how metabolites like NAD+ and ketone bodies directly influence gene expression and inflammation. This is the layer where aging is actively shaped and not just observed. So our guest today, as Raghav mentioned, is Dr. Eric Verdin, President and CEO of the Buck Institute for Research in Aging, the world's first independent institute dedicated entirely to understanding aging and age-related diseases. Eric, welcome to the podcast.
Good to be here with both of you, Maxu and Raghav, and looking forward to the next 45 minutes.
Yeah. Well, Eric, it's amazing to have you here. Honestly, your career has been sort of a guiding part for so many scientists in the aging and longevity and geroscience field. So I actually want to touch a little bit on your career trajectory itself. Your career sort of spans medicine, virology, fundamental of chromatin biology and epigenetic regulation. And then sort of over time, this kind of converged all into focus on aging, specifically like insights on metabolism and metabolic signals and how that regulates gene functions. What really led you to make the shift? Why aging biology and why study it now?
I would love to tell you a very sort of intelligent story of how this was all planned from the beginning. And but one of the beauties of, at least for me, it's always been of an academic career is the ability that you can do whatever you want. And a lot of my career has been driven in some way by chance events, that meetings, interactions with people that really changed the way you look at some things. There was one thing that was clear in terms of my career is that at age 7, I went to a birthday party of a friend and he received a microscope present, and we spent the whole afternoon looking at crystals, copper sulfate crystals and all this. And I still remember this with the same emotion as I did at that time. It was like, oh my God, here's a whole other vision of the world that I had never seen. And from that day, my parents told me that I told everybody I was going to be a scientist.
That's amazing.
Even though I had no clue of what it was, but just this ability to discover, to look at the world in a different way, to imagine what was behind all of these changes. And eventually, I made my way to medical school because of a family friend's advice, who told me you will be a human biologist, and after your training as a physician, you can train in research. And this is what I did. So I did a really long postdoc at Harvard Medical School at the end of medical school, which trained me in research. If you look at my career, it doesn't really make any sense in terms of I went from metabolism, diabetes. As a physician, I was trained in endocrinology and metabolism. So I was very interested in this. I trained with a pretty famous person in the field, Ron Kahn, who initially cloned the insulin receptor and is one of the leaders of our understanding of type 2 diabetes. So that's something that's always been with me. So this fascination with endocrinology and physiology. Ended up working for reasons that are still not clear to my mind, but on HIV transcriptional regulation for many years, for actually close to 20 years. And this work, I worked on this and it was fascinating. We used HIV as a model system to study epigenetic regulation because we knew it was an important aspect. The virus lands as a genetic element in many different parts of the genome, and it has to be transcribed efficiently. So I was fascinated by how does a virus solve a problem like this. And this work led to the cloning and the identification of a series of some of the first epigenetic regulators, the so-called HDACs. We cloned HDAC DAC2 through HDAC7, and eventually then ended up cloning the human equivalent of sirtuins, which had been discovered in yeast. So we cloned 31 to 37 human protein genes. And that all of a sudden, I started reading about aging. And I realized, there was an intersection with a field that I had really missed, which was metabolism, energy, mitochondrial function, and so on. And so that was around 1996. I hate to be reminded of this, close to 30 years ago. And so at that point, I sort of moved back through in some way what had been my original love of interest, which is metabolism, energy metabolism, and so on, linked with epigenetics. And this is where my lab I think has made its mark. It's really trying to understand how do these epigenetic regulators intersect with metabolism. We know metabolism is the source of energy generation or anabolic molecules. So I've always been reasoning that there had to be an incredible tight regulation between gene expression control and metabolism. And so this has led to work on NAD. I'm sure we'll have a chance to talk about this, but also the role of ketones, the role of acetyl-CoA, and so on. And so one area that I'm still fascinated by is really that intersection between small metabolites and how they speak to the epigenetic circuitry.
Yeah,
amazing.
that is
It's a
amazing.
very long answer, but it's sort of
I absolutely love it. I feel like we have so many areas in life that are common. Like myself, I also started, with biology, and then my dad said like you have to do medicine, and then suddenly I didn't know what I wanted to do. I just knew that I wanted to do research, but back in Brazil, it wasn't really possible. End up doing sports science, and then coming to Australia. So I love how it's not a straight line in most cases, and same with Raghav. I'm sure that he will talk about.
It was a straight line.
Yeah.
Like I'm a computational biologist studying aging science, but I started off as a electronics and computer science engineer.
loved it. Loved it.
Yeah, it's
That-
amazing.
that frankly, I tell, it's the best part of life for me. That poetry of the unexpected. If I had met my young colleagues when I was in medical school. One of them struck me. He told me, I asked him, what's your goal in life? He says, I want to publish 100 papers by the age I'm 30, when I'm 30. And I thought, this is the most ridiculous thing I've ever heard. How could you actually define your life on the number of papers? I told him I want to publish one important paper before I'm 30, but not necessarily 100. So I think just allowing sometimes life to carry you and to be surprised by it and to take a road that you don't know where it's going, to me is really the best aspect of what we do.
Absolutely. I love that. Thank you so much for sharing and for some wisdom too. So for our listeners that are new and they haven't heard about the Buck Institute, what do you think it makes it unique in the aging research landscape, and how does it structure bringing together metabolism, the neuroscience, the immunology, and all other areas, and enables this integrated approach to aging?
That's a great question. So I'll just give you a brief introduction. So between 1995, many in your audience probably know, there was a revolution in our understanding of aging by the work of Tom Johnson, Cynthia Kenyon, Lenny Guarente, Gary Rifkin, all identified the first mutation that controlled aging. And this broke sort of a mold that had been imposed on the aging field, that aging was sort of an entropy-driven, sort of random process by which cells would become dysfunctional one at a time. And what people had not appreciated is the importance of genetic circuits behind that, the toxic insults and our ability to regenerate and to repair. And so the identification of selective mutation that controlled the aging process just opened up people's mind to the fact now aging was a tractable molecular system for which you could use all the tools of modern molecular biology, mutations, but also drugs. It opened up the idea if you can actually target with a mutation, you could probably identify some drugs that would target the same protein and interfere with the aging process. So that really is what laid the ground for someone, Mrs. Buck, who left some money to, she passed away and she left the money to a foundation and they wanted to spend it. She wanted to help the aged. That was her mission. And so they decided that rather than, give it to old people, they would actually create an institute that would capitalize on these changes in our understanding of aging and try to benefit humanity. So that's how the Buck was founded. That means we have a small endowment from the Buck Foundation, which I wish would cover all of our work, but only covers about 5% of our work and our budget. So we were started in 1999 as a not-for-profit academic institution. We're located about 30 minutes north of San Francisco. If you're familiar with the Golden Gate Bridge, once you cross it, you drive 30 minutes north and you hit Sonoma, and we're right at the border between Marin County and Sonoma. We live in a beautiful building, which was designed by I.M. Pei, the famous architect who also did the pyramids in the Louvre or the East Wing of the National Gallery. So it's a great building. Some of you might have come here for meetings. It's an inspiring place for me to come and work every day. Beautiful architecture. We have today about 300 different employees coming from 34 different countries. And so we operate like any lab that you would see. So 23 different PIs who each run a little operations. We touch on everything related to aging. That's the only thing that will get you a job at the Buck is you have to work on something that's related to aging. And it goes from the basic biology, studying aging in C. elegans or in fruit flies or in mice or in humans, to people studying the pathogenesis of diseases like Alzheimer's or Parkinson's disease. So I've been here for 9 years. And one of the things, the dimension that I've added is the whole idea of studying humans. And my way of presenting it to our faculty is that we're adding another animal models, which is the human. And so that means some things that are similar to basic research in fruit fly and Drosophila, but some things that are different. For example, we started a clinical research unit. Right now we have eight clinical trials that are ongoing. I think there's a really important aspect of our field. We don't want to just study aging in worms. We want to study it in humans. We want to make a difference in people's health. And so we've recruited also clinical trialists, physicians who know how to do clinical trials. We have a big new emphasis on AI, about 30% of our budget now being devoted to programs that are looking at AI. And I'm happy to discuss all of this. So there's the whole vision of what I want the Buck to do. So we still have 2 more buildings to build. So if one of your listeners has some extra change burning at the bottom of their pockets, there's an amazing opportunity to build 2 more buildings and to really continue expanding. My vision of the Buck when I will retire, hopefully in a long time, is to create a fully integrated institute, probably about double the size of what we have right now, that really spans the whole field of aging research, going from the most simple animal all the way to humans. And with the goal, frankly, of really changing medicine and changing the trajectory of how we age and how long we live and how well we live.
Yeah. I have to say, the vision around the Buck has been amazing, Eric. And how quickly, sort of within a span of 30 years, so much has been done by the Institute to really establish itself as
I'm happy to actually to highlight. I didn't even do this, but I was hoping you were going to get to this, talk about, so what are we known for?
Yeah, I actually, well, let me start by saying the building itself, as you mentioned, is so beautiful. When I visited for the first time, I remember there was like sort of the fog was still kind of covering over the hills and the sun was just kind of rising. And it is just such a beautiful, beautiful sight.
I love the love. It is it is a truly special place. And I was at UCSF and at the Gladstone for 20 years before that. And when I left, I was pretty worried because the Mission Bay campus of UCSF and UCSF in whole has about 20,000 people. And here I was moving to I think multi-billion-dollar organization at UCSF. So many colleagues, all of the equipment. Some colleagues told me, what are you going to do at the Buck with 300 people? You're going to be so isolated. We're on the top of a hill.
Yeah.
But what I have found is exactly the opposite. I think I've one of the things that characterizes us is an incredibly tight community. Everybody is interested in the same thing, in aging. So we have lots of collaborations, lots of discussions. It's an amazing place to work. If you look at our papers, all of them are collaborative. And then you walk into this building every morning. I call it a temple for medicine. I'm not a religious person, but when I walk into this building, I feel the weight of the responsibility of what we do, and to do it in such a beautiful space surrounded by nature is a true privilege.
Absolutely. And actually, maybe on the point of what you were saying, Eric, could you maybe highlight some of the really major impactful things that have happened over 30 years that Buck has been in existence?
Yeah. After these initial papers, the 4 or 5 papers that were characterized, defining mutations that control aging, that's when the initial work was focused on. And so, people like Gordon Lithgow and Simon Melov and others went to work and tried to understand. People did not believe these results first. They thought these are exceptions. These are things that happen to C. elegans. It's not relevant to mice. Well, they went to work and were able to map pathways, and so identify many mutations that control aging. It's actually kind of easy to do this. And I think Gordon Lithgow has told me that in C. elegans alone, there's close to more than 600 genes that will control lifespan in a direct manner or semi-direct manner. So that's what people thought was an exception is not. It's actually quite easy to do. And it's actually quite interesting to see why has an evolution actually favored this? Why do we need to make mutations to make animals live longer? Well, there's a whole series of reasons that we can discuss, but quite often it's because reproduction is favored rather than a long lifespan. So that there's always some kind of antagonism between the two, reproductive ability and reproducing fast versus living long. Lots of amazing biology in this whole area. So that was one contribution. First, just mapping the pathways, the role of insulin signaling, the role of proteostasis. Much of the work of the Buck, frankly, led to the identification of what we call the hallmarks of aging. We were not associated with the paper, but a lot of the work that was done here led to the identification of these sort of pillars of aging, these molecular pathways that contribute to aging. That would be number one. Number two was Gordon Lithgow and other colleagues, Brian Kennedy at the time, came up with the so-called geroscience hypothesis. What they observed as they were playing around with these mutations, they found that when you tinker with aging, the animals live longer, but they also live healthier. And so out of this came the idea that the common trunk for all the chronic disease of aging that we are seeing around in our parents, grandparents, think about heart attacks, stroke, and all of this. That's all driven by aging pathways. If you target aging, you are actually not only making animals live longer, but you suppress the development of all of these diseases. It has a huge potential for medicine. And so the geroscience hypothesis has become something that's been embraced by the NIH, by Felipe Sierra and others, and is really hopefully driving a new way to look at the etiology, the cause of these diseases, and how to interfere with their development. So this would be two things. Number three, actually, Gordon Lithgow also was the first one to identify a drug that increased lifespan. That was published in a science paper, and first time that anyone had actually given a drug to an animal and made it live longer, which was actually quite interesting. Came also the Judy Campisi, who was actually created pretty much one of the whole pillars of aging, the whole idea of senescence. So Judy was a faculty at the Buck, identified the first molecular marker of senescence with senescence-associated beta-gal, and was able to use this to enumerate senescent cells, came up with the whole concept of senolytics, started the first company that targeted senescent cells with the whole idea of senolysis. That was a company called Unity Bioscience, founded this with Arch Venture. And eventually that whole idea permeated the whole institute, so that many of us, including my lab, ended up working on the mechanism of senescence and their relevance to disease. And this is work that's still ongoing. Then more recently, one of the things, as I mentioned, when I came to the Buck, I was struck as a physician by the fact that people were studying aging in these animal models and really people not paying as much attention as I thought they should to humans. And so what was the implication of all of these findings to humans? Are all of these pillars validated in humans and so on? So since I've come, we've recruited a number of people who have an MD background, have better understanding of human biology. We've developed a whole series of organoid model systems. And so right now, what's exciting, we created the clinical research unit where we, as I mentioned, we're doing clinical trials. So we're really trying to go to the next step, which is obviously much harder when you go to humans. How do you take all these discoveries and translate it into human health? So I think that
Do you even measure how long is it going to be
well, the whole
affecting?
biomarkers of aging is really one big area right now that we're working on, which is the next big frontier. How do we translate aging research into humans?
Yeah. And which is
Absolutely.
to say, I think the buck has gotten some really amazing grants in the recent past, including 2 ARPA-H funded grants, which
Yes,
is, I don't think any institute of that sort of, I think, per person production in that so many sort of ARPA-H grants that I have seen, which is amazing.
It's really inspiring, all the breadth of work, like all the big names and discoveries that have been happening in the institute. It's no wonder that you have received such prizes. So
and
yeah.
you ain't seen anything yet. I can tell you without really spilling the beans, we are in the process of recruiting one of the top. We recruited Lee Hood
Yes.
and Nathan Price, who
Yeah.
are arguably two of the best people in the world in terms of bioinformatics, biology, and really studying longevity in humans. We are in the process right now, and I won't spill the beans, but I can tell you when people see who are in the process of recruiting, their jaws are going to drop. So, and that's really for me, it's a dream. Is really I want the Buck to be one of the best places in the world where you can do not only aging research but biology. Period. And I think we're getting there. Right now we have 7 recruitments that are ongoing, so lots of excitement in terms of where the institute is going to be in the next 5 years. And I think to me, I count my blessing every day. I mean, to be at the head of an organization like this is just amazing.
Well done to you too. It really is exciting times. So let me just go back a little bit into one of the points that you didn't really touch on, but I know that is another major area of your research, which is the immune aging. So we often hear the term of inflammaging, the chronic low-grade inflammation that increases with age. So how does metabolism and immune function intersect here? And why is it that the immune system has such a key role into all of the other aging processes and eventually in Healthspan?
Good. So let me answer the first question first, which is why the immune system? Why do I study that? And that was so I've always done immunology in the context of epigenetics, but when I came to the Buck, I wanted to, as I mentioned, really shift the institute towards a more human-centric aspect. And so I showed the good example by shifting my lab to study human immunology. And you can wonder why the role of the immune system in aging. Well, there are a whole series of reasons, which in some way have been validated a posteriori. But I get the bet was the correct bet. The bet was that the immune system and the central nervous systems are the only distributed organs. In a way, when you're talking about the immune system, of course, there's concentration of immune cells in your spleen, in your lymph node, in your thymus, but every tissue is full of immune cells, macrophages, lymphocytes, neutrophils, and so on. So the immune system is constantly patrolling the whole body, checking for pathogens, making sure that there's no tissue to repair, and so on. Plus, the immune system plays a big role in tissue remodeling. So the whole idea was that the immune system has to be one of the major driver of the aging process. And so this has been validated in 2 different papers by Maria Mittelbrunn and Laura Niederhofer. Both of them showed in mice, if you make a mutation only in the immune system, you actually induce aging of the immune system, but that itself induces secondary aging in every other tissue. So these are 2 landmark papers that sort of have validated, at least in mice, that a primary lesion in the immune system will induce organismal aging. That means you better keep your immune system in good shape. The other thing that's been shown more recently is by Tony Whisker and his colleagues, who have generated a series of selective clocks that are organ specific, that allow you to measure how does your immune system versus your brain versus your lungs and so on age. And they measure the rate of each of those. And it's technology that's been commercialized by a company called Vero. Full disclosure, I'm on the board of that company because I think it's really exciting biology. They use that technology to go back to UK Biobank and try to see, okay, which organ aging is predictive of lifespan. And there are two organs, not unexpectedly, it's the brain and the immune system that are the most predictive. So that means if your brain ages faster, you're likely to live shorter. And so, again, this is the prediction based on the fact that even though your brain is in your head, the nerve extremities go absolutely everywhere. Every organ is innervated, every muscle, everything essentially. So if you have lesions or dysfunction in your brain, your whole organism is going to be affected. Now, so that would be the first question in some way. What is it about the immune system? Now, if I can just diverge a little bit to basic immunology, there are two parts of the immune system. There's so-called innate immune system, which is the rapid fire response that is activated as soon as a pathogen enters or as soon as there's tissue damage. It is not specific, but it can recognize bacteria in a non-specific manner. It's called the innate immune system. That system becomes chronically activated, the so-called inflammaging. So it's actually inflammation is the readout. And we know inflammation, which is normally a protective response, if it becomes chronically activated, becomes deleterious. It leads to tissue damage. The other part of the immune system is so-called adaptive immune response, is much more selective. That's the one for which you can generate memory. For example, if you get vaccinated, if you get infected by a bacteria or a virus, eventually your T cells and your B cells are going to recognize the specific molecular signature of that pathogen, and they will be amplified. And so that when the pathogen is eliminated, you will have been so-called primed, so that if that pathogen or the virus comes back, the response is very quick and very protective. So that's the basis for vaccination. Now, it turns out that response actually decreases with aging, versus the innate immune response increases in an abnormal way. The adaptive response decreases, which makes us more prone to virus infection. COVID-19 was a good example. Influenza, RSV, pneumonia, all these diseases really disproportionately kill the elderly. The other thing also is they make us much less efficient at responding to vaccines. So we have a double problem. We cannot respond to the infection in the proper way, or we respond in an inappropriate way. In COVID-19, people showed exaggerated inflammation, so inflammaging was bad. They had inappropriate T cell and B cell response, so that left them unable to respond. So combine all of this together, this is why I think studying and understanding the role of the immune system in the aging process is really, in my opinion, has not been studied enough. And everything that we study has been quite interesting. We can make a lot of brand new discoveries, and I think I would hope that there's going to be a lot more interest in the future in addressing these problems.
Yeah. It is quite exciting, and I do think like inflammaging is such a central focal point of aging. And often I also feel not given enough focus sometimes. We almost sort of it's such a permeance across the body in a way, because it affects every other system. Increased inflammaging leads to increased cardiovascular risk, increased chronic kidney disease risk, increased cognitive risk, everything. Yet it's so often overlooked as this singular mechanism that might be driving aging so significantly. And I really think we need to focus quite more heavily. And actually, a lot of work that you have done in sort of HIV positive models sort of indicates towards that, right? That the low grade inflammation that we see in HIV positive individuals essentially drives a lot of the chronic disease risks that they see, even after they are being sort of treated with a lot of the medications, which is quite astonishing, to be honest. But I
Agreed.
want to talk about interventions for a bit, and especially since you're so excited about sort of the human aspect of the work that's going on at the Buck. I've been sort of following a bit of your LinkedIn posts recently, and I see you've been posting quite exciting and giving your opinions in a lot of way. And one of the things that you have been sort of really giving sort of a balanced optimism for with realism, I would say, the excitement around drugs, right, like rapamycin and metformin, but also the emphasis on lifestyle interventions like exercise, nutrition, and how incredibly powerful they remain. There's a lot of noise in the field, right? How should we think about this balance today? And I know this is a loaded question that I'm asking you, but feel free to.
But I'm really happy to be able to address it because people just love to put you in a box and or to diminish your influence or do. So I've learned all of these games that are being played, and I can tell you frankly, there's a whole group of people in the aging field who don't like me, and so they've called me Dr. Healthspan, and and or I've been called the grumpy doctor of longevity medicine. I mean, call all kinds of names, and I can see it sometimes in the posts how people just love to put you in a box, and I think this really gives me a good chance to put the record straight. So here's really how I think about all this. Today, we don't have any medicine, any supplement that are known to increase lifespan or health span, period. That's the truth. And we might have some. I believe that the GLP-1 agonist and SGLT2 inhibitors and metformin and maybe a whole series of carbos, some other drugs might be really targeting these pathways. But we don't have the demonstration. It's only inferred from data that comes in mice. And I think this is our work to do, is to actually establish the validity, the safety, and the efficacy of these drugs using the gold standards, which is a randomized clinical trial. Okay, so that's really how we have gotten so far in terms of medicine, is by applying the most rigorous standards to demonstrate safety and efficacy. And I think those standards are going to have to be applied to longevity medicine like any other field. And the problem, by the way, is even harder because we're dealing with people who are, in general, sort of healthy. So if we want to increase their lifespan, you better be sure that what you're giving to someone for 20, 30 years is not actually going to make them live shorter or be more sick. Having been trained as a physician, I know how many drugs have side effects. And, I'll give you an example. I know quite a few people who've approached me in their 30s and saying, I want to go on metformin. I said, yeah, why do you want to do this? Well, because I think it's going to make me live longer. So if you consider that it will crater your testosterone, no one's talking about this. Okay, so in a young man, you can really damage your testosterone response with metformin. So I tell people, there's not such a thing as a free lunch. Many medicine have untoward side effects, and we have to be very careful. So that's the aspect. I'm as excited as anybody is, and I should, because we sit on the top of all of this amazing data in animal models, that at some point we will be able to generate drugs that increase lifespan and health span. That's the future, and we're working on it. We're doing clinical trials, so some people call me, they said I lack ambition because I will never tell you that I'm going to live to 140. Okay. Some of my colleagues are doing this. I think it's deplorable. It should not be done because they have no evidence that this is going to happen. Some people are raising the ante. Some supplement guys is telling everybody is going to live to 170. I could walk around and scream on the top of every roof that I'm going to live to 180. It's total BS. I mean, they don't have any evidence for making these claims. And I think it irritates me because I think it does not speak to seriousness. It's a hype machine. It's part of what I call Instagram medicine. It just, I'm going to scream louder than you are, and my number is bigger than yours, and therefore I'm more representative. So that's my beef, is I really hate this hype aspect of our work. Let's just walk before we run. Let's project reasonable expectations for the population. Because the thing that bothers me the most is that speaking about living forever is one of the oldest dreams of humanity for everybody, not just for those people, for me too. I want to live old and happy and healthy, but at the same time, I recognize the limitations of what I'm doing right now. But I am ambitious. don't misunderstand the realism on the conservatism from ambition, because we are working, actually, we're doing more clinical trials than many of these other people who are walking around making these crazy predictions. So that's number one. Number two is we know things today that will increase your health span and your lifespan. And I think we have to speak about those. But it's not one or the other. It's not health span versus lifespan. The two, I think, are really intricately linked. But while we're working, to changing the world for the next 20, 30, 40 years, I think to me, it seems that as a responsible physician, I have to let people know what can they do today. And this is the whole health span thing. I'm convinced today that most people could live to 90, 95 in good health if they were to implement everything that we know. And so a lot of our effort right now is to try to define these so-called lifestyle factors. Think about nutrition, physical activity, sleep, stress mitigation, and finally, the soft things like a sense of purpose or loving connections and so on are all what the tools that we have today. We sort of know about them in general terms, but we have not studied them molecularly. For example, now with all of these measuring clocks that we can use, which form of exercise is the most conducive to longevity? Right now, everybody exercises sort of willy-nilly thinking, if you listen to some people, they will tell you, well, everything is in muscle strength, everything is muscle mass. Some of the people will tell you, no, it's all zone 2. None of them know for sure what is really the best regimen for the physical activity. I would like to know, so I don't know, so I do a little bit of everything. I try to have a bit of a higher VO2 max, I lift some weights, but frankly, I'm navigating all of this in the dark. Now with tools of biomarkers, I think we can start doing what my colleague here at the Buck, Lee Hood and Nathan Price have called scientific wellness. So it's applying the clocks and the molecular tools to understand what is it about nutrition, what is it about sleep, what is it about exercise that really allows you to live longer, and bring all of this soft science, the wellness sort of industry, to a molecular understanding and to a rational design. So that's really one area that I think we cannot ignore this. There's 20 to 30 years of extra lifespan and health span that we can grab today, because we know that people who have a great habits tend to live longer than others. So I hope that sort of reconciles the two aspects that I think where the field should be going. It's not one or the other. It's not being crazy about prediction versus ignoring what we can do today.
I think you're absolutely primarily because I think like the intervention at some level know that works is calorie restriction, right? And that's the one lifestyle intervention that has been tested in these settings or and seem to help with slowing down the rates of aging. We don't really actually even know a lot of these therapeutics or even some of the biohacking mechanisms people are trying to work. Someone used this terminology the other day and called it longevity entertainment, and it really
I call it Instagram medicine.
yeah, and I completely second that. At some level, it is that, right? When you're perpetuating these ideas and ideologies without sort of rooted science.
Exactly.
Yeah, it is not useful.
And I love that you touched on the exercise part as well, because very recent we had another invited speaker in the podcast, and he was actually mentioning how the decline in muscle mass due to lack of exercise is one of the signals that can say that your brain is going to decline 10 years prior to the declining brain. And as you just mentioned, the two only organ systems that seem to drive aging so fast is when you have the brain being affected or the immune system. So they just show, how exercise somehow is affecting all of this. And not to mention that it does also affect your immune system and resilience and so on and so forth. So definitely we have to do the things that we know that work. And I'm not saying that none of those supplements or medications don't work, but we don't simply have the measures. And to measure how much is actually going to affect in a human life, it's going to take years. And I feel like we're just trying to predict everything and just say that it's going to happen the same way that it happened with animal models, and it's not actually the truth. So I think we just need to slow down. It's gonna come, but we just have to stay well until then, and do the things that will allow us to stay well until then. So yeah, I
I
fully agree
agree.
with that as well.
My argument to people is, the science will come. I would say in the next 5 to 10 years, we will have the first demonstration that some of these interventions, drugs or supplements, actually are really moving everything in the right direction. We already have some hints, but you want to position yourself so that you are as healthy as possible 10 years from now to benefit from the next wave. And I think that's really, for me, is the, you cannot imagine that these drugs coming on the background of someone living unhealthy are going to be very helpful. And I think I tell everyone who wants to listen that try to live as healthy as you can today so that you will benefit from the discoveries that will come in the future. And
Yeah. Yeah. Actually, on the point of future, looking 10 years ahead, right? There's so much work that is happening in different fields right now, right? There's metabolism, of course, there's epigenetics, there's immune regulation, and of course, there's so many other dimensions to sort of the aging field that is sort of ongoing research. What are the most exciting developments that you see emerging right now, the convergence around this? Uh, what you what are the things that you're really willing to take your bet?
it's very hard to make a bet on what's going to emerge. I still think if I look at the biology in a simple organism, the pathways that we have mapped, it seems that the insulin and glucose pathway, so our ability to metabolize carbohydrate via insulin, is one of the major pathways that emerges from fruit flies, from C. elegans, from mice, and now in a completely independent manner from the ITP program, where people have been testing which human drugs might be increasing lifespan. It turns out many of these drugs that I've mentioned earlier, SGLT2 inhibitors, metformin in some animals, acarbose, maybe GLP-1 agonists, are all targeting insulin and glucose metabolism and signaling. If I had to put my money on something that's going to happen in the next 10 years, is that one of those is going to emerge with a very strong response in humans. So that's really from the metabolism point of view. And we still do not fully understand how this works. It's an incredibly complex system. As I mentioned, I did my postdoc in the lab of Ron Kahn at the Joslin Clinic at Harvard. He spent his whole life trying to understand insulin signaling. Nir Barzilai has done a lot of beautiful work in this as well. So I think it's really, to me, this is one area that I think is really important and not fully understood, so it needs more work. The other area that really excites me, and I think it excites everybody in the aging field, is the whole Yamanaka factor reprogramming.
Yeah, for sure.
So problem there is the fact that I think the findings are real. We can really rewind the clock. We can rewind the clock all the way back to a primary embryonic stem cells, which is unbelievable. We can also partially rewind it to a more youthful state. And so, Juan Carlos Ipsisua Belmonte, a whole Altos company, I mean, they've been like the true pioneers of really doing all of this. David Sinclair now has joined the fray with his company, looking at this in the eye. So there's many people studying this, which is a remarkable process. The problem is, as a rejuvenation strategy for an organism, no one has been able to explain to me how you're going to be doing this. Now, maybe the only thing you need, because the problem is these are vectors, they're, and so you need to be able to transduce all cells, or at least a majority. Maybe it would be sufficient to do it in the immune system. So I haven't heard anybody actually really come up so far with a mechanism how this is going to be done to increase longevity. But that being said, it can be used in a more sort of isolated manner, the same way life science, David Sinclair's company, is going after doing it in the eye, for example, for glaucoma or some other unique condition. So I think maybe but this is not longevity. And there's an ocean separating the idea of doing this in the eye to doing it in the whole organ. And so which means it's not a problem of longevity, it becomes a problem of vectors, signals transduction of drugs, and so on. So, and this is where it's going to be rate limiting. That being said, maybe you just need to do it in the brain, or you just need to do it in the immune system. So I try to stay optimistic because I think the effect that have been seen by many are so dramatic that it has to give you a pause and saying maybe we've discovered, Belmonte has really discovered something pretty unique. Now, the only thing about this whole story is I've been kicking myself for the last 20 years because when I was at the Gladstone, when Shinya Yamanaka made these discoveries, he was the floor below me, and I was already working on aging, and I was already thinking at the time, there has to be a meaning related to aging. But I never had the courage to actually do the experiment, and I should have. But anyway, that's history. Sometimes it tells you when you have a hint or sort of the impression that you should be going after something. I think I did. I didn't, and someone else did, which is good for the field, but not good for me.
You could have won the Nobel Prize with him.
Well, I don't know. Nobody's won the Nobel Prize for this yet because and the reason is, and Shinya did, but the reason being that everyone recognizes the difficulty of bringing this to the level of an organism.
Mm-hmm. Yeah, no, that absolutely. I think it's a very inspiring area, the epigenetic reprogramming. And the only thing that I always think about is how sometimes reprogramming or reversing, to a young state, in my mind, it just goes straight into like equals to cancer because somehow we can't handle, having all this growth as children do. So how do we actually balance it out? And I still have my, sort of like worries about it in a way. But
you're not
absolutely.
the only one. You're not the only one. Remember, they had quite a few tumors initially. In the case of the one that's being done in the eye, I believe they remove c-Myc, which is one of the major drivers. But one should still, one of the properties of stem cell is to proliferate. So there's always the possibility that some of these cells are going to start proliferating.
Yeah, absolutely. Okay, so as a final question, this has been absolutely wonderful and an absolute pleasure to have you here. But we have been asking this recently to all of our guests, and we would love to hear from you. In your opinion, who do you think has really been moving this field in incredible ways? I know that you mentioned so many people already during this episode, but who do you think we should invite next to come and give their view on aging and their perspective, and someone that is really making an impact right now, in your opinion?
And doesn't have to be one. It could be more.
It doesn't have to be one.
It doesn't have to be one. It can be more than one. But we typically think of like always taking our guest opinion on who should we invite next.
All right, I'll pitch one of my colleagues, although, I'm probably going to make some enemies of some other colleagues that I haven't pitched, but I'll send you the list. I would say one person that is really impressing me a lot right now is Nathan Price. And if you haven't read his book called The Age of Scientific Wellness, he wrote this book with Lee Hood. And I think it's going to be a really, another person would be Lee Hood. He's also a colleague at the Buck. He is the father of something called P4 medicine, the idea that medicine can be precision, predictive, participatory, and there's one more that I always forget. So this idea of reinventing medicine based on what we understand about aging science, it's coming of age today. So maybe you can have the two of them together. I think they really are helping us at the Buck formulate what this new vision of modern aging medicine will look like in the future. And I think they will provide for entertainment. And Lee also on top of it has such an amazing history in terms of everything he's discovered. You might know that he invented the first DNA sequencing machine. He was behind the sequencing of the human genome. So I think they both are incredible. So I would recommend both of them.
Lee is definitely a stalwart. He is amazing. And I've heard Nathan's talk at a couple of conferences, and his visions of how to implement AI in the field of longevity is beautiful. Those would be amazing guests.
Happy to make the two introductions.
Yes, for sure.
Amazing. There we
Well,
go.
thank you so much, Eric, for joining us today. We will link a bunch of resources from Eric's work, as well as, of course, the Buck Institute, which has been doing some really amazing work over the last sort of 2-3 decades. And to our listeners who enjoyed our conversation today, please follow Decoding Longevity and share it with your network and leave us a review. Until next time, thank you.
And come and follow me on LinkedIn. Verdin Lab actually is started a Substack where we are actually doing unique papers in the literature, and I'm really giving our sort of analysis of these papers. I think it's think about it as a journal club for the general public, and
Really
it's
like that
actually a
idea.
lot of fun. So we started posting.
Yeah, we'll link your LinkedIn, but more importantly, your Substack. I really like this idea of like a public
Mm,
journal club.
Yeah,
Uh,
that's
yeah, it's
it's
very cool.
amazing.
Yeah,
okay.
Mm.
All
cool.
right.
Thank you so much, Eric. Really enjoyed
Thank
this.
you
Good
so
to
much,
see you
Eric.
all. Thank you.
Thank you.